Hello, everybody, and welcome to the Bat Advent Calendar. My name is Thomas Hille. I'm currently a postdoc at the University of Hohenheim in Germany. Today, I got the opportunity to tell you something about bats, but I won't be talking only about bats. I will also be talking about bat flies. For those of you who have worked with bats, you might already know what I'm talking about and probably have seen them in the field. And for all of those of you who never have heard of them, bat flies are probably the most conspicuous ectoparasites of bats. Bat flies are small, so between 1 to 5 millimeter in size. Some of them look like tiny spiders, that they're running around on the wings of the bat or they're diving through their fur. So they are highly host-specific blood-sucking insects, and often this is associated with only a single host species. So they are really dependent on their bat host. But they're not only dependent on their bat host, they're also dependent on the roosting structure the bat is using. So bat flies are reproducing by adenotrophic viviparity. This means a fertilized egg is hatching inside the female fly, followed by 3 larval stages, which are carried inside the female and nourished by an intrauterine accessory gland. So the third instar larvae is finally deposited on a suitable substrate somewhere in the bat's roost. This can be a cave wall. And after around 3 to 4 weeks, an adult fly emerges and is searching for a new bat host. So we know that their host, the bats, are heavily affected by forest fragmentation and changing environments. In recent years, it has been shown that this effect is species or feeding guild dependent. Highly specialized forest bat species, for example, are strongly negatively affected and even go locally extinct, while some more generalist bat species might even profit and become more abundant as they can cope better with these changing environments. As you heard before, these bat parasites, these bat flies, they are highly dependent on their host species. So the question we are asking in the paper I will present you today was, how do these bat flies react to changing environments? To investigate this, we were capturing bats in central Panama. We had 14 sample sites in continuous forested islands in the middle of the Panama Canal and forest fragments immersed in agricultural landscape. We captured over 8,000 bats there. In the study, however, I will just focus on the 7 most commonly captured species. We were correlating the prevalence and also the intensity of parasite infestations with variables on landscape level. So for example, type of the surrounding matrix and habitat measures, on variables on host species level, like the host abundance and the roosting ecology, and variables on host individual level, which would be like host sex or the reproductive status. I will spare you all the exact statistics and results here and jump directly into the main findings. So we found a clear density dependency. This means the more hosts are available, the more parasites. This makes totally sense, right? And we also found that the roosting ecology, so the preference for a specific type of roost, namely a leaf tent or a hollow tree or a cave, has an effect on parasitism. Here we found the more stable the roosting environment is, the more likely the bat is parasitized. If you recall, bat flies need the bat roost to deposit their pupae. So the more stable a roost, the more stable are also the environmental conditions for these bat flies and favorable for their development. If we have a look at habitat fragmentation, we found host species specific reactions therefore. So we did not find a consistent pattern over all our focus species. We identified different landscape and habitat variables depending on host species. So if we just pick out one example to explain what we found. Let's have a look at Artivius jamaicensis. I will call them from now on HAs. That's much shorter and easier to pronounce, to say. So HAs are very common frugivorous bats, and we captured them in pretty the same relative abundance in all of our habitat types, in all of our 3 habitat types we were sampling. We expected to see higher levels of parasitization in forest fragments compared to continuous forest sites, as habitat fragmentation often affects the health of inhabiting animals, and which makes them vulnerable to parasite infections. However, in HAs, we saw a lower level of parasitization in fragments surrounded by agricultural matrix when we compared them to our continuous forest sites. This was really surprising, as we expected this to be the reverse. So why do these HAs have less parasites in these forest fragments? So when we remember, bed flies need the bed roost to successfully reproduce. However, in these fragments, we have a quite high anthropogenic influence. So large trees in which you normally find these hollows for roosting are often cut down for timber production. So these important roosts are missing for both for beds and for bed flies. On the other hand, HAs are really flexible in roost selection. So we can find them roosting in caves, in hollow trees, or even openly in the foliage. And therefore, we suspect that the beds in these fragments are using predominantly roost in the foliage. As you can imagine, roosting in the foliage is not really desirable if you're a bed fly, as this is not a safe, favorable environment for your development. Microclimatic conditions there are quite different, and you have predators you normally won't find in a hollow tree. So this together might lead to the lower numbers of parasites we have observed in these fragments. Now, looking at this bed species, the HAs, we do not see any difference in abundance in these different forest fragments compared to our continuous protected forest sites. They find food and roosting possibilities, and they are thriving there. However, the associated bed flies, they are really heavily affected. So given the current speed of the environmental change and the loss of habitat, some of these bed fly species might even be at risk of extinction. While we are aware that the habitat loss affects the local bed community, these beds themselves are a habitat to a quite intriguing parasite community. So I would like to highlight that we still do not know enough about the life history, for example, the roosting ecology of many of our bed species. And we know even less about their associated parasites. And if you now think what a crazy system this bed fly system is, just wait for it. These bed ectoparasites, these bed flies, they can be parasitized themselves by tiny ectoparasitic fungi called Labulveniales, forming a trophic Christmas tree. And with this, I will end here and wish you happy holidays.